What is transport across the cell membrane? It's the movement of particles down the gradient called diffusion. A gradient is when something is unbalanced in a concentration. Question 2. What is hypotonic solution? It is a solution that has less solute and more water than another solution. Question number 3. What is a hypertonic solution? It is a solution that has more solute and less water than another solution. Question 4. What are the characteristics of the cell membrane of animal cells? It is semi-permeable that have two primary functions. First, forming an outer boundary on the cell and hold the cell together. Second, allowing certain substances to pass through the cell membrane and prohibiting others. Question 5. What molecules can pass freely across the cell membrane? Oxygen can pass because it is a small molecule, so it can easily pass through the cell membrane. Carbon dioxide is also a small molecule, so it can also be able to pass through the cell membrane. The materials that we used for this project were 2 eggs, 500 milliliters of acetic acid, one 1 liter bottle, one scale, 250 to 500 milliliter transparent beakers, 500 milliliters of water, 500 milliliters of corn syrup, camera for photos or video, and a computer to put the information recorded. Before we began the project, we put the 2 eggs in a 1 liter flask and add vinegar to cover them. We let them stay like that for 3 days. The purpose of this step was to get rid of the hard calcium layer surrounding the eggs, thus exposing the semi-permeable membrane so that we can work with them. After 3 days, we remove the eggs from the vinegar and rinse them with water. Now the eggs have an exposed semi-permeable membrane. After that, we weigh the eggs and record them weight in a table. The first weight of the egg was 75 grams and the second weight of the egg was 70 grams. For the next step, we put 1 egg in 1 flask and fill it with water, which is a hypotonic solution. In the other egg, we put it in another flask filled with corn syrup, which is a hypertonic solution. For this step, both eggs should be completely covered with their respective solutions. We let the eggs for 8 hours and revise them every 2 to 3 hours to see any change. After the 8 hours, we remove the eggs from the solutions and weigh them. We record them in the table. The first egg, which was in a hypotonic solution, weighted 75 grams. The second egg that was in a hypertonic solution weighted 37 grams. Now we will change the solution for each egg. The egg that was in a hypertonic solution now will be in a hypotonic solution, and vice versa. We are going to let the eggs for the same 8 hours to see the changes. We're going to take a video every 2 or 3 hours to see the changes. After 8 hours, we remove the eggs from the solutions and weigh them. We record their weight in a table. The egg that now is in a hypertonic solution weighs 30 grams, and the second egg that was in a hypertonic solution and now in a hypotonic solution weighs 95 grams. We conclude that when an egg is in a hypertonic solution, the egg's weight will increase, because in a hypertonic solution, water enters and fills the central back wall of the cell, pressing it into the cell wall. And when an egg is in a hypertonic solution, the egg's weight will decrease, because in a hypertonic solution, water inside the cell moves out of the cell, causing the cell to shrink.